I now wish to focus on longitudinal profiles of skeletal muscle fibres. This cross-section through the tongue of a rabbit demonstrates skeletal muscle. It's been stained by the Masson technique to distinguish the muscle fibres from connective tissue. A layer of stratified squamous epithelium, stained reddish-orange, surrounds the tongue. The dorsal surface is modified with projections of papillae that I discuss in my lecture on the tongue. The green-blue ring under this epithelium is loose, irregular connective tissue, called the lamina propria. Loose connective tissue stained green also surrounds bundles of skeletal muscle fibres, which are stained red. The individual fibres are large enough to be seen easily at this low magnification and are much larger than those of the other cells in the green connective tissue. You can't even see those cells. Even the fat cells are dwarfed by skeletal muscle fibres, and this is because a fibre is not a true cell, but a syncytium formed by the fusion of hundreds or thousands of cells called myoblasts into a giant multinuclear structure surrounded by a continuous cell membrane. Because the fibres are arranged so orderly, you can easily find ones cut transversely, longitudinally, and obliquely. They can be up to about 100 micrometers in diameter. In fibres running longitudinally, you can see the characteristic cross striations of skeletal muscle fibres nicely. In fibres cut transversely, note the locations of the nuclei just under the cell membrane, which is too fine to be resolved at this magnification. Transverse sections also show the rod-like myofibrils in cross-section within the fibres, and these give the fibres their grainy texture. Myofibrils are the structural and functional subunits of a muscle fibre. Look very carefully at sections cut longitudinally here and here, and note the cross bands or striations show up particularly well. This banding reflects the A and I banding pattern in sarcomeres due to the regular arrangement of thick and thin myofilaments organised in each myofibril. And again, note the number of nuclei on this fibre sectioned longitudinally. Some are slightly out of focus because they're below the focal plane and perhaps on the other side of the cylindrical shaped muscle fibre. A small proportion of these nuclei adjacent to the fibre just under the external lamina that I'll describe later are satellite cells. They're a reserve progenitor cell or reserve myoblast, capable of dividing and producing new muscle fibres following muscle injury, although the regenerative capacity of skeletal muscle is limited, especially after major trauma to the muscle. And just in case I forget to mention this later, cardiac muscle cells don't have satellite cells associated with them, so have no regenerative capacity beyond early childhood. Damage to cardiac muscle results in the region being replaced by fibroblasts and connective tissue, forming a myocardial scar. But regeneration readily occurs in smooth muscle because the cells or fibres are small and less differentiated.